So, good evening, all of you. We are going to discuss this acid-base balance chapter. I will discuss in detail, beta, because anyhow it's a lengthy chapter, and we'll discuss every aspect of this chapter. Anyhow, it's an essay question. See here in this chapter. Now, basically, I told you previously also, like the acid-base balance introduction, like the normal pH of our body. How much is the normal pH? It is 7.4, isn't it?
Yes, ma'am.
So we have various mechanisms in our body which are actually not letting this pH to change. So we have blood buffers, we have respiratory mechanisms, and we have renal mechanism. So all these mechanisms will work in such a way that they will resist alteration in pH. The pH is not allowed to change. Okay. Now, before we go to the details of this buffer, we should know the definition of buffer. What do you mean by buffer? A buffer is defined as solution of weak acid and its salt with strong base. Solution of weak acid and its salt with strong base. So that means whenever this combination is there, whenever acidity is increasing in the blood, the base component of this buffer will actually neutralize the acidity. Whenever alkalinity is increasing, then the acid component of buffer will neutralize the acidity. Okay, so that is the meaning of buffer. So basically, in order to maintain the blood pH, we have three mechanisms: blood buffers, respiratory mechanism, and renal mechanism. Now, when you talk about blood buffers, we have three components in the blood which will allow the pH to be maintained always 7.4. So what are the three buffer systems? We have bicarbonate buffer, phosphate buffer, and protein buffer. When we were discussing plasma proteins, one of the functions of plasma proteins you read is plasma proteins help in buffering, isn't it? Now see here, bicarbonate buffer system. It is sodium bicarbonate and carbonic acid combination that is called as bicarbonate buffer. Sodium bicarbonate and carbonic acid. You should remember this name and also formula. The combination of sodium bicarbonate and carbonic acid is bicarbonate buffer, and this is the predominant buffer which is found in our blood. Okay, so extracellular fluid is nothing but in the blood. 90% of our buffer system comes from this sodium bicarbonate and carbonic acid. So it is found specially in extracellular fluid, that is plasma. So basically we know this formula. How is this? Yesterday when I was doing chloride bicarbonate shift, then also I told you H2O plus CO2 combines to form H2CO3, isn't it? So that means basically water is combining with carbon dioxide to form this carbonic acid, which is highly unstable, and it is splitting into hydrogen plus bicarbonate ions. So we have a very simple derivative here, which you are supposed to write because it's always an essay question. Regulation of blood pH, and one of the mechanisms will be asked in detail, and most of the times the mechanism which is always asked in detail is blood buffers. So under blood buffers, we have to give the description of all three. So we are discussing about bicarbonate. So this is like law of mass action at equilibrium, which is represented as Ka. This is dissociation constant of carbonic acid. So here what you are writing, H plus HCO3 minus divided by H2CO3. Okay, this is left hand and this is right hand reactants. No, beta? These are reactants and these are products. So products are written in the numerator and reactant is written in the denominator. Now you are just rewriting this with cross multiplication. Okay, so hydrogen is brought to this side, this Ka is coming this side, H2CO3 is going up and HCO3 minus is coming down. This is simple mass beta where you are doing the cross multiplication method, that's it. Then now we know that pH is nothing but log 1 by H. So here what you are doing is log 1 by H you are now applying is equal to log 1 by Ka. So instead of this Ka, what you are writing is log 1 by Ka plus log this one. H2CO3, HCO3. Now you are writing in reverse direction because everything is taken, like numerator position is all coming in denominator's position. H has become 1 by H plus, no, that's why Ka, which was in numerator, it has become 1 by Ka. This has gone to numerators and position numerator into denominators. Very simple math. You are just taking the reciprocals and logarithms for logs. So now we know, like log 1 by Ka, this one is nothing but pKa. Okay, so don't think much, just try to practice and memorize. We are not doing maths anyhow. Okay, so 1 by Ka is, instead of writing log 1 by Ka, you can write pKa beta. We already know log 1 by H is nothing but pH. Okay, instead of log 1 by H plus, you are writing pH. Instead of log 1 by Ka, you are writing pKa. This remains same. Log bicarbonate by uh, like H2CO3. Okay, then the above equation is valid for any buffer pair. General equation, that means above one HCO3 is base and H2CO3 is acid. Okay, so generally it is evident from this equation that pH is dependent on the ratio of concentration of base to acid. Now, after writing all these, basically in our blood, base concentration will be actually more when you compare with acid. Okay, so because most of the times whatever are produced in our body are acids only. For example, we have ketone bodies. What are ketone bodies? Acetone, acetoacetic acid, beta hydroxybutyric acid. That means every time acids are only produced. Even if you just check the glycolysis cycle, we have lactic acid, pyruvic acid, isn't it? So that means acid production is more in the body. In order to compensate that, we have base is more in the blood when you compare with acid, which is almost 24 times more when you compare with the acid. Okay, so that is to neutralize many acids which are formed during metabolic activities. So the plasma bicarbonate concentration is 24 millimoles, okay, when you compare with acid composition. Okay, so again there is a simple, again whatever values are given here, they are actually substituted in this formula. Then we know pH is 7.4, pKa is taken as 6.1, log bicarbonate is nothing but base. No, base is how much? 24. How much is acid? That is 1.2. So once you simplify 1.2, it comes to 20, no, if this point goes up. So log 20 is nothing but 1.3. So 6.1 plus 1.3 comes to 7.4. So of course, though this is cumbersome or though it is very boring or whatever, you just practice this because blood buffers is very very important. Bicarbonate buffer system, this is. Okay, so I told you bicarbonate to carbonic acid, that means base by acid. Base is how much times more? I said 20 times more than acid. That's what is called as alkali reserve. In our body, all acids are only produced. In order to neutralize, we have more base in the blood in the form of buffers, so that it can immediately neutralize the acids which are being formed. Hence you use the word alkali reserve. So after that we have phosphate buffer system. Okay, so in phosphate buffer system we have sodium dihydrogen phosphate and disodium hydrogen phosphate. How many buffers we had? Three, no? We are done with bicarbonate in the blood. Second is phosphate buffer. Third will be protein, plasma protein buffer. So phosphate buffer, you should know the again composition: sodium dihydrogen phosphate and disodium hydrogen phosphate combination is phosphate buffer. I told you this bicarbonate buffer is rich in plasma, that is extracellular fluid, whereas phosphate buffer is rich in within the cell, intracellular buffer. This is. Had it been in the blood, it should have been much more powerful than bicarbonate buffer, but basically it is present inside the cells, not outside the cells. Hence, inside the cells, the pH is always maintained constant with the help of phosphate buffer system. Last but not least, what we have in blood is protein buffer. So what are plasma proteins? We have albumin, globulin, prothrombin, fibrinogen. So among that, inside the RBC also we have hemoglobin. So all these plasma proteins and hemoglobin together, they help in the regulation of blood pH. Hence they form protein buffer system. Okay. So that was our first system which helps the maintenance of blood pH. Once you know the maintenance of blood pH by blood buffers, then we have respiratory mechanisms. That means lung is also helping. Lung is also not allowing the pH to deviate, either increase or decrease beyond 7.4. So how the lung is working? So this mechanism is, yesterday we have studied something like chloride bicarbonate shift. That's what we have studied in physiology. In fact, the same thing comes here. See, this is during the exhalation of carbon dioxide. Basically, when carbon dioxide levels are increasing in blood, what happens? Carbon dioxide combines with water to form carbonic acid. Hence, carbonic acid levels will increase in blood in case of carbon dioxide retention. Yesterday, we have studied forced respiratory volume, timed vital capacity when we were doing physiology. In such respiratory disorders, you will see expiration. That means during first second, and third second, the respiration is reduced. So in such cases, carbon dioxide is retained in the blood. It is not allowed to move into the atmosphere due to obstructive and restrictive diseases of lungs. So in such case, when carbon dioxide levels are rising, it combines with water to form carbonic acid. But somehow the lung is trying to compensate by hyperventilation. What do you mean by hyperventilation? The normal rate of breathing is 12 to 14 per minute. But in case of hyperventilation, the breathing rate becomes faster. It may go up to 20 to 25 breaths per minute, which will wash out excessive carbon dioxide. That is one of the protective mechanism. We have respiratory center in the brain. We have studied, no, apneustic center, pneumotaxic center, dorsal group of neurons, ventral group of neurons. So these centers are getting stimulated, and it will cause the rate and depth of respiration is increased, okay, through which the carbon dioxide is washed out. So that is how the lungs are also helping in regulation of body pH. Okay, that is respiratory mechanism for pH regulation. So here you can just draw this picture. Inside the lung we have in the pulmonary arteries and veins we have RBCs, and this is plasma, that is blood. So from the blood, excessive carbon dioxide has now got transported to the RBCs in the lungs. So this carbon dioxide is combining with water in the presence of carbonic anhydrase. Here you have the name, carbonic anhydrase, to form H2CO3, that is carbonic acid. We don't want this acid, right? Hence, this acid is now splitting into hydrogen ion and bicarbonate ion. So, this hydrogen ion combines with hemoglobin to form HHB, okay, hydroxyhemoglobin, and this bicarbonate is now transported to the plasma. We always need bicarbonate, alkali reserve. How much is more alkali? 20 times more when you compare with acid. So bicarbonate is transported to the blood so that blood can neutralize more and more acids, isn't it? In exchange with chloride. You can also call it as chloride bicarbonate shift. Okay. So that is how lungs are regulating. I told you we have three mechanisms, right? Blood over, lungs are also respiratory mechanism over. Then last but not least, we have renal mechanism. Kidneys also play a major role in regulation of pH. Let us see how. It's also very easy. See here, in the tubular cells, I have shown you the structure of nephron many times. We have Bowman's capsule, proximal convoluted tubule, Henle's loop, distal convoluted tubule, and collecting duct. So in the tubular cells, what is happening? There are certain ionic changes happening. So inside the tubular lumen, hydrogen ions will be secreted and excreted. I'll show you diagrammatically. Bicarbonate will be reabsorbed by these tubular cells into blood circulation. This is alkali reserve. Even kidney is also helping in alkali reserve. Then excretion of titratable acid, then excretion of ammonium ions. Okay, these are the four mechanisms we have. Okay, now see here, first we have renal regulation of blood pH. First mechanism, what do we have? Excretion of hydrogen ions. So in the excretion of hydrogen ions, you are finding what is that? See, in the tubular lumen, that is inside the tubular cell of nephron, the universal formula always you remember. Carbon dioxide combines with water to form H2CO3 in the presence of carbonic anhydrase. H2CO3 splits into H plus and HCO3 minus. This bicarbonate enters into the blood and it is saved there, okay, as alkali reserve. We do not want hydrogen. Acid, go away. Hence, this acid is now going into the tubular lumen. It is combining with some base to form a salt which is excreted in urine. Okay, so that is one mechanism, that is excretion of hydrogen ions. Reabsorption of bicarbonate. Actually, we need not draw one more picture. Here also you can see the bicarbonate is reabsorbed into blood circulation. But still there is a one more figure has been shown. You can also draw the same thing. Here you are finding reabsorption of bicarbonate. Again, same universal formula you have to write. Inside the tubular cell, the carbon dioxide and water are combining to form carbonic acid in the presence of carbonic anhydrase. It splits into hydrogen and bicarbonate. Bicarbonate is coming into the blood. Okay, here hydrogen is going into the tubular lumen. He has specified now like this is combining with HCO3 minus to form H2CO3. Okay, so it is again coming back to the tubular cell for this type of mechanism. Okay, where bicarbonate is coming back to the blood. Just draw this picture, like understand it and write in your own words. Finally, you are seeing there is reabsorption of bicarbonate. What is the third mechanism? I said excretion of acids, titratable acids. I'll show you what is that. See here, this is excretion of acids. Same formula again inside the tubular cell. You will see the formation of H2CO3, same thing. Only thing is now this hydrogen ion, when it is entering into the lumen, it is combining with sodium dihydrogen phosphate to form a titratable acid which is excreted in the urine. The pH of the tubular fluid was 7.4. Because now this acid is excreted, urinary pH is becoming acidic. But blood pH is only 7.4. We need blood pH to be maintained constant. Okay, our body is excreting acids. That's how urine is acidic. So what is the last mechanism the kidney is doing? See, excretion of ammonium ions. Ammonium ions also can cause very dangerous. I told you ammonia, if it is increased in blood, it can cause coma. So hence ammonium ions also should be excreted. Again, same thing. You see carbon dioxide, water, H2O, this is coming and getting like stored in the blood as alkali reserve. And we know that sodium from the lumen is now entering into the blood. So from the protein breakdown, which ammonia, we have seen transamination, deamination, I have explained you in my yesterday's lecture, I remember. This ammonia which is set free, it is now combining with this hydrogen acid to form ammonia. NH3 is ammonia, H plus ion is NH4, ammonium salt. It is excreted in urine. So these are the four mechanisms which kidney is helping us with to get rid of hydrogen ions, to get rid of acids. Okay, that's how these are the three mechanisms. You just need to think cool and just sit and practice all these and write in your own words. Okay, so blood buffers, respiratory mechanisms, and renal mechanisms. Okay. So once you know the buffer mechanism, we have to know, actually we have discussed metabolic acidos, metabolic and Respiratory alkalosis already in our previous class. Now see here, major clinical causes of acid base disorders. We have four acid base disorders. Wherever you get this word acidosis, what does it mean? The blood acidity is increasing. So metabolic acidosis is due to decrease in bicarbonate. Obviously, if bicarbonate levels are increased, hydrogen ions level will increase, which will cause the acidity. The causes may be diabetes mellitus, where we have ketoacidosis. That means acetoacetic acid, beta hydroxybutyric acid, and acetone levels are increased, leading to metabolic acidosis. Okay, renal failure, lactic acidosis. Lactic acidosis will occur if the person is doing heavy exercise, okay, where lactic acid is getting accumulated in the muscles. Severe diarrhea cases where lot of bicarbonate is lost in loose motions. Okay, in renal tubular acidosis, that means the kidneys are actually absorbing more hydrogen ions, and hydrogen ions in the blood are actually increasing. So all these conditions will lead to metabolic acidosis. In case of respiratory acidosis, there is increase in carbonic acid. H2CO3 levels in the blood are increased. It may be due to respiratory disorders like asthma, pneumonia, heart attack, cardiac arrest. There may be obstruction in airways, chest deformities. Respiratory center of medulla oblongata is not working because person has consumed certain drugs, depressive drugs like opium, opiates. Okay, so metabolic alkalosis and respiratory alkalosis are the conditions like this. I already told you, metabolic alkalosis is due to increase in bicarbonate, respiratory is due to decrease in carbonic acid, and these are the conditions. Okay, so this is for your essay answer. So what is quite big, so I have told you everything. Then gout also we did. What is gout? I gave you the treatment also. Gout is excessive breakdown of purines, which is leading to excessive production of uric acid. That is primary gout. And we have also discussed the treatment. What is the treatment? Allopurinol. Allopurinol is the drug of choice, okay, which prevents the conversion of hypoxanthine to xanthine. From there, you will see uric acid is not produced. Hence, we can prevent gout, isn't it? Then gastric function test, I will tell you. Organ function test is the chapter. See here we have under organ function test, we have tests like gastric function test. Okay. So what is this gastric function test? What is the meaning? See here we are using Ryle's tube. This is renal, no? Creatinine clearance test is also there. C is equal to U into V by P. Just a minute. So for gastric function test, why do we do this test? To check if gastric juice is composition is normal or not, if there is any blood or if there is any cancer cells, if there are any cancer cells in the stomach, in the small intestines. Okay, we generally use gastric analysis. So in case of gastric analysis, there will be a tube available which is called as Ryle's tube. So generally, we will ask the patient to come empty stomach early in the morning, and we generally insert the tube, that Ryle's tube, to pass. We have certain markings, graduated markings on the Ryle's tube. If you want to collect things from the stomach, you will insert to 1 point. If you want till duodenum, you will insert to one more marking. So from whichever part of the GIT you want, you just insert the Ryle's tube and you aspirate. Pump out the gastric contents, pump out the intestinal contents, and collect it in a container. Then that whatever is being collected is analyzed. That's what is called as gastric analysis, intestinal analysis. For example, if you are doing gastric analysis, you are checking if HCl concentration is correct or not, if all enzymes are there or not, is there any blood tinge, are you finding any cancer cells, any further investigation is required. You will come to know whether it is duodenum, whether it is jejunum ileum, whether it is stomach. Based on the area where you have stopped your Ryle's tube and you have collected the contents, you will give the report. That's what is called as gastric analysis, also called as gastric function test. Okay, once you are getting something like blood tinge is there, it can be gastric ulcer. But if you are finding cancer cells also, then that means there may be some gastric tumor. So in order to actually like rule out, we will do further tests, like we have to go with endoscopy, where you will take a biopsy of, biopsy is a piece of tissue is taken from whichever area is suspected. Then that will be studied pathologically to check if any cancer cells are there. Like histology slide we prepare, no, in pathology also we prepare the same slide to check if normal cells are there or cancer cells are there. Okay, so this is the meaning of gastric function test. So in order to increase the gastric juices, we give certain medicines, like we can give alcohol also. Okay, we can give certain pentagastrin injection also. We can give certain things are there which will increase the secretion of gastric juice. Okay, that's what is called as gastric function test. So so that is what a histamine also we can give, so as to increase the production of gastric juice. Read once, it's very interesting, you will easily understand. Creatinine clearance test is repeated. Structure of plasma membrane. Meanwhile, Niharika, just take the photograph of functions of copper, and when I ask you,
Yes,
you
ma'am.
please share it on the screen. Please don't share till I tell you, please. Okay.
Yes, ma'am.
I am taking you to plasma membrane structure. So, what are the basic functions of copper? Copper containing enzymes are there, no, like ceruloplasmin. We have studied ceruloplasmin when we were doing absorption of iron. So, one of the chief functions of copper is it is helping in the transport and absorption of iron, isn't it? That is one function. Copper, if it is increased in the blood and it starts getting deposited in the liver, we have Wilson's disease. Plasma membrane and check. Mm, if you are ready, you can share better. It's taking time for me to check the page number. Niharika.
Yes, ma'am. Should I share now?
Please, you can proceed.
Yes, ma'am.
Given or not, I Thank you so much, beta. See, functions of copper. So copper is essential component of several enzymes like cytochrome oxidase, catalase, tyrosinase. Only few examples are enough, beta. You need not give such a big list. 3 or 4 enzymes are also fine. So copper is necessary for the synthesis of hemoglobin. Okay, then it is also very important for the conversion of certain lysine residues. That means it helps, you just write, it helps in the formation of collagen and elastin proteins. Okay, so ceruloplasmin, I told you, it serves as a ferroxidase and it is involved in the conversion of iron from ferrous form to ferric form. Okay, I told you, no, it is helping in the transportation of iron in the plasma. It is necessary for the synthesis of melanin pigments and also phospholipids. So copper helps in the development of bones and nervous system. So copper containing non-enzymatic proteins have been identified, although their functions are not clearly known. Like you can write few names. Copper containing non-enzymatic proteins are hepatocuprin is there. We don't know the functions also, but names are there. Then cerebrocuprin and all. Okay. Then dietary requirements is not asked, beta. Okay, that is functions only is asked. Plasma membrane chapter from last page. Okay, you can stop sharing, beta. So structure of plasma membrane is actually found in the last chapters. It may take little time. Singer-Nicholson's model. Do you remember, beta? Plasma protein, bilipid layer, proteins are studded in between. It is highly semi-permeable. It is allowing only few things which are useful for this. Can anyone share the better answer? Not finding. Maybe I have missed. Now our question is plasma membrane, isn't it? So when you talk about plasma membrane, we should know like this plasma membrane as cell membrane, like we have fluid mosaic model. So which shows like it is bilipid layer. That means you are finding these green balls, okay, with these filaments black in color. These are all, this is all bilipid layer. In between this bilipid layer, whatever pink areas are shown, these are all proteins. So proteins are studied in this bilipid layer. Okay, so that is like fluid mosaic model. We have like plasma membrane lipid bilayer originally proposed for membrane structure by Davison and Daniel, but the most accepted one we have is fluid mosaic model, which was proposed by Singer-Nicholson. Okay, the biological membrane usually it has thickness of 5 to 8 nanometers.